In this lesson, I will be talking about the first of the molecular events of DNA replication, which is initiation, some vocabularies and enzymes related to initiation are as follows. I'm not going to be talking about them individually right now, but I will explain them as we go along. Before a DNA can be copied, several things need to happen that allow for effective copying or effective replication. Um, Initiator proteins sort of hold a DNA molecule down, and then a specific group of enzymes called helicase start unwinding or unzipping the hydrogen bonds that hold the complementary strands together. Um, this basically where the helicase starts. Its unzipping is basically the point of origin, which is a specific nucleotide sequence. As as the helicase moves along the DNA, it creates a replication bubble, which grows in opposite directions. As the replication bubble keeps on growing, it creates two Y-shaped regions called the replication forks, moving in opposite directions. Now, the total DNA inside each human cell laid end to end from each chromosome is about 2 meters long. And in each DNA molecule, there may be one specific or several simultaneous replication bubble happening at once. As the replication fork moves up or down the DNA, it also, as you can see, this is in a replication fork, the Y-shaped region that we talked about earlier. As it grows, it creates those tension in the adjacent areas in the DNA, which must be relieved. And they're relieved by a group of enzymes called topoisomerase 2. As topoisomerase 2 relieves the tension, another set of enzymes get to work called the single-stranded binding proteins. Now, each exposed DNA strand is very it's susceptible to coiling back on itself as it is unstable, and it is held stable by the SSB, or the single strand binding proteins, which basically helps it from coiling back on itself. So this is basically the main events, or the main these are basically the main um, events of replication, after which each strand can then be blah blah blah.